Also, you're muted in case if you're saying something, I wouldn't know.
Yes.
Okay.
I feel like this is my first Zoom, but of course it's not. Here we go. So can you?
Yeah, I see.
And things are moving as well?
Yes, absolutely. That's perfect.
So I'm going first then, I guess, right? Is that correct?
Yes, yeah. You get the privilege because you're junior.
That's fine. That's good.
Okay. I think it's 11. So I just wanted, before we introduce our wonderful speakers today, I just wanted to introduce Martina. So we are going to have Dr. Martina Lersch help us as well. She did her PhD in Joanna Iwaska's lab. So she has been around in the cell migration for a while. And she's now a postdoc with Dr. Claire Waterman at NIH, where again, she's working on aspects of cell migration. She loves integrants. So if you have any integrant related stuff you ever want to ask, I would really direct you to Martina. Please don't come to me. I know nothing about integrants. Okay, with that, we really have the pleasure of two wonderful speakers today, Abdel and Alba. And we will first go with Abdel, and Martina will introduce today.
So hi everyone, my name is Martina. I'm really happy to be here. So Ankita kindly asked me to join this awesome series and to help out every now and then if there will be use for third person. And today I'm pleasure to introduce to Abdel. So Abdel holds a PhD in mechanical engineering from McMaster University in Canada. And in 2019, he obtained a Research Foundation Flanders postdoctoral fellowship to develop methods to apply mechanical stresses to organoids and study the relation between cell patterning and forces at KU Leuven in Belgium in the lab of Adrian Ranga. He became intrigued with the extracellular matrix dynamics and went on to study its relationship with morphogenesis. And right now he is a postdoctoral fellow at CEMM, Research Center for Molecular Medicine of the Austrian Academy of Sciences. And he plans to investigate the metabolic dimension of fate mechanoregulation. So Abdel, with that, the floor is yours.
Thank you very much. So thank you very much for this very nice introduction, and I'm very happy to be here. And I want to tell you a little bit about the extracellular matrix and epithelial morphogenesis. So the question that we're kind of intrigued with is really how biology takes shape. So if we look at from a developmental point of view, so if we look at development, we can see that through the progression of time, very distinct biologies start to emerge. And what is fascinating about this is they all have very different shapes. And what is even more fascinating about this is that shapes all have originated from essentially one simpler shape. We can see that biology became very good at actually building shapes. And the study of shapes in biology perhaps have started maybe 150 years ago. It hasn't been, it's not so long ago that the notion that shapes can in turn define biology started to take place. So here we can see, for example, that cell shape can be a driver of neuralization, and this is a very important process in the development of the central nervous system, particularly here in the neural tube closure, let's say. So because shapes emerge from tissue deformations, and tissue deformations are the results of forces, we can start to imagine a mechanobiological dialogue happening between biology and shape of biology. That is part of the mechanism that ensures the right cell is emerging at the right place and at the right time. We became very good at actually deciphering this mechanical conversation by leveraging model systems that primarily, in this case, derive from human pluripotent stem cells, where we can apply biochemical and mechanical cues and then watch what happens essentially, and observe fate specification patterning and morphogenesis, and in that way explore what is the mechanobiological link. So if you could, for example, if you take intestinal organoids and find them in a particular shape, you will be able to see and define mechanical niches for stem cells. Similarly, if you take organoids, in this particular case, human neural tube organoids, and you stretch them, we will be able to find patterning in these organoids, specifically of the floor plate domain, which is important for neural development. So these tools, we're starting to get a better understanding about how multicellular decision-making is taking place in a tissue that is undergoing morphogenesis. We've been primarily occupied by looking at cell patterning, cell shape, cell specification as well, and not so much focusing on the extracellular component of that. So, and tissues, of course, are cellular and extracellular. So we were asking what is the matrix component and what is happening to the ECM as morphogenesis is taking place. And we can see some very interesting and intriguing observations from, again, in development at the onset of gastrulation, for example, during the formation of the primitive streak. There is very high flows of the extracellular matrix that are in line with the cellular flows. So they parallel the cellular flows. And this helps drive and essentially localize the primitive streak formation. Also later in development during axis elongation, for example, here we're looking at fibronectin in green, and then what we in red, and then what we see is that there's also again flow of the extracellular matrix during axis elongation, and this helps direct or chaperone cells towards their correct destination. Even further still in development, again, in central nervous system development, where in the ventral neurocrest pathways, there again is extracellular flows that help direct cells towards their correct destination again. So the example tells us that there's very important cooperation happening between cells in the extracellular matrix during morphogenesis that helps define the functional shapes. And it's not like we don't know this. We already know this from very nice examples in vitro, where when you add, for example, matrigel, which is a natural EPM, to neuroepithelial tissue, we can get these striking observations of morphogenesis, in this case, neurotube folding and closure. But also, if we look at somatogenesis, we can replicate that as well in vitro by gastruloids in a matrigel environment. So this is very all interesting, and despite the fact that we have some, let's say, we're starting to decipher the relationship between cell collective behavior and ECM. In this case, for example, in this nice study from last year, where they have avian skin patterning that is in a reciprocal relationship between the substrate. We still don't have very good understanding about this relationship in epithelium. And so for us, it's what we kind of wanted to look at. And we decided to look at it from a very sort of the onset of shape of biology, at the onset of morphogenesis, essentially. And that is concerning gastrulation. So we're looking again here at the primitive streak, because we know that there are ECM flow occurring at this stage, but it is also coupled with morphogenesis or linked to morphogenesis. So we asked, what is the role of this ECM morphogenesis relationship at the onset of gastrulation? To do this, what we've done is that we took HPSCs, pluripotent stem cells, which mimic sort of the epiblast stage of the embryo. And we started with confluent monolayers of these, and we have a reporter line that is reporting for tight junction. So this is O1. So what you see here is a normal configuration of epithelium, and we decided to see how these cells would react when you subject them to extracellular matrix from the top. We added extracellular matrix to the growth medium, and this is all happening in pluripotent media. And then what we started seeing is these going to have structures that starts to form, which we call them domes, because they are indeed more elevated than the surrounding monolayer. They have a very distinct zone configuration or expression, and this started just spontaneously happening. But what was more interesting is that what happens to the matrigel. To look at matrigel, we stained for laminin, a major component in matrigel, and we saw that it just is accumulated around the domes. Very odd, because we introduced matrigel in a homogeneous fashion. So how did it come here? So we tagged matrigel components using fluorescent beads. And what we saw was this. Striking recruitment, very dramatic recruitment of the extracellular matrix or the matrigel towards domes of morphogenesis or these morphing domes, right? And this is not an isolated event. This was happening everywhere a dome was forming. We get this characteristic recruitment of the extracellular matrix towards the dome. This is at the cell surface, but of course, this is happening in 3D. So we can see here, this is sort of a 3D rendering of these domes or of these what we call toroids of matrigel forming. Again, from a side view, you can clearly see that the matrigel is being highest speed is really coming from the bottom of the matrigel, which is the cell surface, and is being directed towards these domes. And I can tell you right now that this is not a spontaneous event happening because of the matrigel in the well, for example, just spontaneously happening because when there are no cells, we don't find it. So ECM flow is actually a cell-driven and a surface phenomenon, right? So how does it start? How does the flow start? So what we saw is that before dome formation, there is actually a symmetry breaking in the configuration of dome 1. And when this happens, it happens and then it's maintained over 24 hours. So this is in a matrigel condition. The flow doesn't actually start until later. You can start seeing some accumulation here, but it doesn't full-on flow until maybe between hour 6 and 12. That's when you start seeing the heaviest accumulations. And then we were thinking that perhaps it is cell migration towards towards the domes. And I'm sorry to say that even though I'm in the cell migration seminar, it's not cell migration. So it is actually what we see is that if we track single components of this ACM, we can see it very just passing over cells that are relatively remaining stationary here, and it's going in the direction of this symmetry breaking event here, right here. And the velocities of the cell, if we measure the cell flow, let's say the cell velocities, we see them that they're not actually increasing, they're in fact a little bit decreasing, and the ECM flow quickly overtakes that. So cell motion that we observe which is quite stochastic, doesn't really contribute to the directed ECM flow that we observe. So flow is triggered by symmetry breaking events, and we know that. And so we ask the question whether this relationship is bidirectional somehow. And then we also ask, how is global organization achieved? And we ask that because we know that we get periodicity and periodic events happening at the same time. So we were wondering whether this somehow is organized by the relationship between ECM and morphogenesis. So to get better understanding, we need to characterize flow a little bit better. And so we were able to take images like this, or videos essentially, extract the flow field from these images by looking at, by basically doing critical image velocimetry analysis. And then we found that working with divergence of the flow field, and it is allowed to say divergence in this case because we treat the matrigel as a compressible sort of fluid. So we get an image that looks like this, and that's the divergence of the field. And so here, what's in yellow is regions where the flow is diverging or going away from these regions, from everything that's yellow essentially. And in blue are regions where the flow is going towards, right? So naturally the regions where you would find the polarity is changing, but also where the ECM is also accumulating as well. And so we figure out that there's an anti-correlation between polarity change and the divergence of the flow. So we were able to pull these in a minimal kind of in silico model, and we chose sort of a reaction diffusion-like model because of the periodicity that we see. And what we can do with this model is that we can feed it tissue geometries that are based on our in vitro observations, and run the simulations and see what happens where we get domes and where we get these rings, for example. And we see that it actually quite a certain degree recapitulates what we see in vitro. We can also play around with the geometries quite a bit and then we can see that if we change the geometries, we can through a variation of different expressions of the polarity signal in this case in the simulations, which corresponds to ZO1, let's say the ZO1 expression. And we can see again that we can recapitulate this in vitro. One thing about the in silico model is that we can also do perturbation. So we can perturb the model in silico first by looking at the flow components. We have two things in the model. We have the flow and the polarity. It seems like what the in silico model tells us, if we perturb the flow or eliminate the divergence essentially of the flow, so in essence not allow the ECM to accumulate somewhere, you will not have any domes, and you will not have this kind of morphogenesis taking places. And indeed, this is exactly what we see. And to recapitulate, to do this in vitro, we put our culture system on a shaker, and this is what we see. We see that there are no domes forming. Polarity, we used something that we've used before, by impairing contractility. We know can also influence polarity. And what the in silico model tells us is that you will get essentially one continuous dome morphology. So you're not going to have domes that are separated and periodic. You'll just have a phenotype that is just dome everywhere. And this is exactly also what we saw. So we abrogated the monolayer phenotype from our culture when we added blattostatium. So we can see that morphogenesis does form a bidirectional relationship. By coupling these two components together, we're able to recapitulate what we see in vitro, and so we can say that there is a bidirectional relationship, and this somehow can link smaller local events, what's happening at the single dome to global scales. What is actually driving ECM flow? This is something that we still didn't know, and to do that, we looked at the one dome level and analyzed the flow in these domes. We looked at high magnification, and what we saw again is that we were able to get these flow fields, but we saw that the flow is not necessarily very homogeneous. It happens in bursts, and it happens it doesn't happen in very homogeneously around the dome. And we were able to link the flow field to cells that are at the periphery of the dome that are undergoing some polarity changes. So we can see that when there's a higher polarity here, the flow is high essentially in magnitude, and then as this one dies out, another cell down there is changing its polarity, and then from the zone expression, and then this allows the flow to also increase again. And here you can see it not in the video format, but it might be a little easier to interpret. So that was quite interesting, but it doesn't tell us what is actually engaging the ECM. Right? We know that the ECM must be the surface of the cell, something that has to be engaging with the ECM. So one thing that we decided to do is to look at the surface of our cells, and we used electron microscopy in this case, and we saw a drastic change in the cell morphology, surface morphology, essentially, in particular the microvilli in this case, where in the control condition we found these microvilli to be quite sparse and stochastic in configuration, whereas when you put the matrigel, they become much denser, number one, and there seemed to be some alignment, and the alignment perhaps is actually aligned towards the dome direction. So that prompts us to actually look at this process in a dynamic way, and we're able to look at microvilli dynamics by looking at actin dynamics, which couples to the microvilli. And we saw that there's coherent motion of microvilli dynamics towards dome locations at the cells that are at the periphery of these domes. So it seems like you can think about it as maybe a conveyor belt on the cells that are actually pulling cells towards the domes, putting the ECM towards the domes. And the velocities also of these microvilli are quite consistent with what we see in terms of ECM velocity. So the ECM is triggered symmetry breaking events. It's driven by some polarity changes and is actually required for morphogenesis, but something that we haven't answered yet is what about these cell fates, right? So what happens to the fates of the cells when in this whole process? And to answer this, we looked at, we employed single cell RNA sequencing, and we saw from the get-go a big difference in terms of identity in control and matricial condition. And we mapped our data to the human gastrula data set, and we found, not surprisingly, most of them are epiblast cells or resemble epiblast. But what was interesting is that when you add matrigel, there seems to be some, there's actually promote primitive streak-like identity. And we can see a reliance more on nodal, which didn't make sense for us because of the primitive streak dependence on that, rather than BMP, which happens to be in the control phase. So we looked for early PS marker, early primitive streak markers, and we found that NANOG actually, which is a potency marker, but in this case, it's also an early primitive streak marker, and we can see that it is actually quite located. It's located in the domes, and it's highest in the domes, right? And this, we also found that this. From literature, so definitely missing the reference here, but from literature, the NANOG again is actually gets localized in the region where at the early primitive streak, and later it's lost at the expense of mercury. So we decided to look further into this matrigel identity, and we were able to find three major identities or three major groups in the matrigel data set, based on the NANOG expression from low to high, and we did some trajectory analysis on this, and found that what we believe would be corresponding to dome and monolayer phenotypes. In this case, we have one major dome cluster, let's say, or group, and then the other two groups are monolayer groups. And what caught our eye is that one of in the dome group, let's say, this is where NANOG is highest, and this is what we expected because we already saw this. The monolayer group, one of them is quite really SOX2 high, and we did the staining for this. We saw that indeed SOX2 is kind of not really so much in the domes, but it's in the monolayer surrounding the domes, whereas the domes, in order to gain the NANOG expression and be in the dome, you technically have to lose your SOX2 identity. And so perhaps we're witnessing some sort of also posterior anterior axis definition here in the dome being posterior, where the primitive streak usually would take place, and then monolayer in the anterior part. So the most frustrating part is that this streak-like phenotype is actually we're lacking completely mercury. So when we stain for mercury, we don't actually find it. So we saw is that, like I said, NANOG is high in the domes. We don't have mercury markers, so it is only a PS-like morphology. But our hypothesis is that we're lacking certain factors to push the system to adopt the mercury fate, and that with the right factors, perhaps the domes would then actually acquire that fate. Indeed, this is what we did. So when we did the mesoderm differentiation in this case, we found that these zones then adopt the mercury fate at the expense of NANOG. So this is basically also what happens, what we know happens in vivo, that NANOG is first there, and then it gets lost, and then mercury then takes over, and essentially that's basically what we see. So perhaps the ECM flow morphogenesis somehow biases PS-like phenotype. This process can actually only go so far before you start needing the factors in order to push the system to actually it primes it for that fate, right? So in summary, really, I wanted to say that we can see that there's a morphological event that initiates ECM flow. and then through cell surface processes and polarity changes, ECM flow is being driven and being directed, in particular in a very distinct direction. But then this flow it helps accumulate EM in a particular region closer to the morphogenic event or morphological event, and this helps reinforce and maintain that morphology. And this, in a sense, act as a positive feedback for morphology, for morphogenesis. And then this whole relationship then subsequently defines the transcriptomic landscape and then also morphology landscape of the tissue. And so yeah, I mean, this is basically the story that we have right now, that's I would very much like to open the discussion at this point. And before that, I would like to also thank the lab at Leuven, of course, everyone that participated, yeah, Francesca, Lorenzo, Adrian, of course, and then the lab also in Austria. And a special thanks to Bertrand and Miriam from CBI Toulouse that have participated in many discussions about this. So, and of course, the funding agencies. Yeah, so I guess we'll open it up for questions.
Thank you so much, Abdel. There's actually a question from Anna while other people chime in, and I guess Jen wants to go next. So Anna is asking, did the cells that accumulate on the top of each other, or they lose apical basal polarization with redistribution of, polarity proteins like PAR6 and integrins? Have you looked at after blocking integrins and see if ECM flow stops?
Yeah, no, we haven't blocked any integrins. We do know from let's say focal adhesion staining, so Paxillin, Vinklin, that these tend to be lower in the dome region, right? But laminin receptors, for example, are very high at the dome regions in the regions that are actually facing the matrigel. So in the matrigel, in the media. So we believe that there is, of course, a very big contribution for integrins in this case in terms of actual anchoring. Right? We're not sure if this is actually what is.
So what Anna was also asking was also the apical basal polarity. Is that preserved, or is there changes in that as well?
Yeah, no, there's definitely changes, right? And in a lot of cases, we think polarity is indeed flipped in the domes, right? So there's a flip, so meaning that the apical is no longer in the media facing surface, but rather in the plastic facing surface, right? And this is really because of cells are trying to reinterpret the mechanical microenvironment.
I think Jen can go.
Great, thanks. So just to make sure I was understanding the setup correctly, you are adding the matrigel to the apical surface of the cells, correct?
Yeah.
Abdel?
Mm-hmm.
Yeah,
that's correct.
Okay. I was just wondering about the I guess the sort of the choice to do that, because you wouldn't expect that polarized epithelial cells would be seeing any basement membrane proteins from their apical surface. And so was this coming from a place where you just wanted to give them a natural environment and kind of perturb them and see what they would do, or is there sort of a specific in vivo motivation for that kind of initial setup?
So there's certainly an in vivo motivation as well. Is that we believe it's not necessarily just an entire apical phenomena that's happening. It's more like that there's a change in the polarity that drives the ECM flow. But the other thing is that we're trying to answer a question as to why morphogenesis starts to take place even in vitro when many researchers just add matrigel to micropatterns or any types of other systems. They technically there's a lot of typical exposure, especially in micropattern, right? And this was one of the biggest motivations of the study, is that how the cells start to interpret this signal, right? And yeah, so that's, I don't know if I answered the question. So I mean, for sure, when you add matrigel to micropattern, let's say epithelium, which then undergoes a lot of morphogenesis, you do see the exposure will be at the apical side, right? So, and we wanted to understand how the cells deal with that.
Yeah, no, that does answer my question. Thank you very much.
Okay, then we have another question in the chat box. So how do organelles respond to mechanical cues? Do they change their location and function?
How do organelles? Is that...
Yes.
Well, I haven't, I mean, if by organelles you mean like inside the cells, or like I guess there must be some mechanical response. We haven't really investigated this so much. I mean, we know for sure that there's some like subcellular polarity that takes place as well, right? So you get accumulation of certain proteins in the cell due to mechanical forces. So so it's possible that this is also playing a role in that.
Yeah, thank you. And then we have a raised hand by Sophie Ashraf. So Sophie, please go ahead and unmute yourself.
Oh, hi. I have a question. So I'm not convinced that ECM is not a passive passenger. So it goes where cells go, rather than influencing morphogenesis. So I have two questions for you then. So you said that you observe a bidirectional relationship between morphogenesis and ECM flow, but besides computational evidence, what is the experimental evidence, and how did you uncouple flow from anything else? And the second question, it's in the same sort of theme, is you said that ECM flow is required for morphogenesis. So what is the evidence for that as well? Thanks.
Yeah, so I think so. What I didn't show you is ECM. We've done some other experiments using other ECM components, right? So turns out that when you, for example, PEG, right? And we worked a lot with PEG, so polyethylene glycol, it's a linear elastic material. You don't get any sort of this morphogenesis. So, and there's a very good reason for that, is that it can really flow, right? And even if you spike PEG with matrigel at the same concentration as the media, you still don't get flow, right? So even so, the components of the matrigel alone, they don't trigger morphogenesis. You need to have a redistribution of your matrigel components so that you have regions that will be high in matrigel, regions that will be low in matrigel, right?
I'm sorry,
so
but how do you block that redistribution? I'm sorry, I didn't understand that.
How do you block redistribution?
Yeah, how do you block a redistribution?
Yeah, so polyethylene glycol is a sort of a synthetic matrix, right? And it forms a mesh, let's say, on top of your cells. So just like matrigel would make, it can't be not really be remodeled.
Also, you mix polyethylene glycol with matrigel, and matrigel doesn't.
Well, matrigel, yeah, because matrigel then it's quite stuck, right? So the components of the matrigel is there, and we know that because we did scene for it, so we can see that matrigel is in the PEG. But that alone is not enough to trigger morphogenesis. So you definitely need, and if you prevent, of course, the shaking experiments, if you prevent the accumulation of matrigel, the components are still there, right? They do not have the capacity to settle anywhere. And that prevents, again, domes from forming. So,
thank you, Sophie. Sorry, I will have to interrupt
Yeah.
because of the time, but you can
Yeah.
ask offline. So we only have time for one more question. Sorry, Alba, I'm going to take that. And maybe in the end, we have time and Abdel can also answer more questions later on. So I'll quickly take Lara's question, Abdel, but please feel free to go through the chat because there's some more questions. So Lara's asking if in your scheme, in the organization, you have 2 layers of cells, like there's a 1 layer and the bottom layer. And is that because of there's some sort of proliferation involved, or is it just cellular rearrangement?
No, we for sure we have proliferation, and proliferation is higher in the domes. So so there's thickening of these domes first without proliferation, and then over time we start seeing proliferation taking place much more than the monolayer region.
Awesome. Thank you, Abdel. I'm so sorry, Alba. Abdel will take your question later, and I missed some other questions as well. Okay, while Alba puts her slides up, Abdel, if you can put yours down.
Yes.
Thank you. Thank you so much for such a wonderful talk. It's a really different topic, which I usually don't think about. And I also have questions. So if I don't get to you, I'll also email you the questions.
Sure.
Okay. Because of time, we're going to move forward to Dr. Alba Diz-Muñoz. It's really a privilege to introduce her today. She received her PhD in Max Planck Institute of Cell Biology and Genetics in Dresden, Germany. And then she moved to States to do her postdoctoral research in UC Berkeley and UCSF. In 2016, Alba established her own lab at EMBL Heidelberg on mechanobiology of the cell surface. So for a while, Alba's research has been focused on to achieve understanding of how various mechanical layers of cell periphery crosstalk to complex behaviors such as cell migration and differentiation. And I think she is going to talk about membrane curvature and immune cell function, but I would also say that you can go back and look at amazing amount of work which Alba has done on membrane to cortex attachment over the years. So with that, I would like to welcome Alba. Thank you so much for accepting our invitation, and we're really looking forward to your talk today.
Thank you, Ankita. Thank you for your very, very kind introduction. Yes, indeed, it's been a pleasure to join you today. And so I want to thank the organizers for giving me a shout out. I am terrible at keeping up with all the seminar series that are out there. So it's good to be nudged once in a while. And so indeed, we work on the cell surface of immune cells, but also of mouse embryonic stem cells, of fibroblasts. And so today I will be mainly focusing on immune cell migration. But as Ankita said, we are really passionate about membrane to cortex attachment. So if any of you wants to nudge me offline, please do so. And so I want to start my talk today by giving a broad introduction and a little bit of like being in awe with how cool nature is and how powerful it is. And so I want to bring this message where we have an incredible diversity of shapes and forms, but actually nature has very little components. This is if we compare it, for example, with chemical synthesis. And so one of the tricks that nature plays very, very often is actually to recycle the same components in different topologies by using what are called biological composites. So basically you mix together different components with different geometries, and in such a way you obtain things like morphogenesis, but also radical changes in mechanical properties. And so to bring this concept of a composite a little bit closer to home, let's focus for a second on cellulose. So cellulose is the component of the bark of trees, but is also the component of a cotton ball. And so all of you are very familiar with how different mechanically these two are. And so the trick within the bark of tree is actually that cellulose generates these little macrofibrils that are basically arranged in between a bed of lining and hemicellulose. And this is very much resembling what happens with rebar within cement, right? So concrete is very strong under compression, but has very low tensile strength. And so rebar significantly increases this tensile strength in the structure. It's exactly the same happening with cellulose in the bark of trees. And so it's not only with fibers that you can make composite materials, you can also make what we call composite interfaces. And so here the concept is the same, but in this case, you are layering materials. And so if they have different mechanical properties and you apply force, this will inherently lead to morphogenesis. And so to bring this concept a little bit closer to home, let's for 1 second focus on the intestine. And so all of you in the audience are going to have more or less, I'm kind of averaging that we all have the same age, more or less. All of you are going to have the same amount of gut coils. And mechanically, this is like ultra beautiful because it's basically the result of differential growth between the two tissues. Basically, you have a tube, which is your intestine, that is held together by the mesentery. And just because the tube grows faster than the mesentery, the only possible mathematical solution is coiling. And so, as I said, this changes over with age, but once you reach adulthood, all of us are going to have the same amount of coils with the same dimensions, except, as Jennifer might have noticed lately, pregnancy does change things. But this is a topic for another day. And so now let's focus on one of these coils, and let's look actually at what nature does to increase surface area, right? We need to absorb all these nutrients. We already have a long enough intestine. We don't want to make it longer. And so to increase the absorptive area, what we have are actually villi, right? It's these little protrusions that are very beautifully recapitulated in organoids. And so the way that these villi are formed is, again, a mechanical valence. So you have muscle layers that are covering this tube in different orientations. And the piling up of those forces automatically leads to morphogenesis. And so if we go one length scale smaller, right, we now focus on one of these villi. The only way that you can get shape changes is actually if your ECM is compliant enough. The epithelial cells are trying to make the same amount of force. If the ECM is not compliant, you basically are flat. And so this, what I really want to bring forward is this idea that you can use cool mechanics at composite interfaces to generate any shape form you want. And this is not only true in tissues, this is also true within cells. So if we now focus on the animal cell surface for 1 second, we see that it has the plasma membrane, a lipid bilayer, the actomyosin cortex, a very thin cytoskeletal network underneath it, and a layer of things that glues this stuff together, which is what we call membrane to cortex attachment linker proteins. And so this is also a composite interface, and there's also really cool mechanics happening. And one added layer of complexity actually is that there are feedback mechanisms between these different mechanical components. And so this is really what the lab is about. And so what I want to show you today is actually that this balance of forces at the cell periphery generates an absolutely incredible topography. So what you see here is the automatic segmentation of the plasma membrane of an immune cell that we captured with focus ion beam scanning electron microscopy. So we have isotropic resolution in the order of like 5 to 10 nanometers. And what you can see is that the topography of this surface is insane, right? These balance of forces at any length scale you want are going to generate the coolest of morphogenesis. And so the question that we really wanted to address is this just the balance of forces at the periphery, or does it actually encode information? Is this something that the cell uses to feedback into these other mechanical layers at the periphery? And so this work was really pioneered by an absolutely spectacular PhD student in my lab, Eva Sitarska, who's now doing a postdoc in the US at Harvard. And so she really spearheaded this project from the beginning to the end. And so all credit goes to her. And so Eva was really puzzled by immune cell migration and the fact that those cells are absolutely incredible at evading obstacles, right? So there's a couple of movies here from other people that have shown how within organisms, in this case, either a zebrafish larva or the skin of a mouse ear, actually these immune cells, upon damage or even without damage, they actually do these absolutely beautiful dances around within tissues. And so if we now look carefully at those tissues are absolutely full of obstacles, right? We just heard a talk about ECM, the mechanical properties to get out of an endothelial vessel, out of a blood vessel into whatever tissue you want are going to be dramatically changed. And so we wanted to address how they do this. And so when we now focus on one of these cells migrating, what we see is actually that they are extremely dynamic. So what you see here is an HL60 cell. These are the model organisms that we use. And so you can differentiate them, thermally differentiate them in vitro, and then you can put them here in a 2D substrate, and they will do this, right? And so these lamellipodia, they migrate by actin-based polymerization in opposition to bleb-based migration. So they polymerize actin at the leading edge. This allows them to move, but also generates this incredible topography. And so, as I mentioned, we wanted to understand, is this generating information or not? And so one of the families that was a very high candidate in our list were actually bar domain proteins. And so for those of you that are not familiar, this is a family of proteins that have a bar domain. And these are basically banana-shaped domains that can go either in one direction or the other, so binding to inward or to outward curved membranes. And they're very interesting because in vitro, they can actually generate, well, actually also in vivo, they can actually sense or generate curvature in a concentration-dependent manner. And so not only they bind to membranes or they generate curvature, they actually have a whole bunch of other plethora of domains that actually allows them to regulate the cytoskeleton in really interesting ways. And so Eva set off to try and figure out what's the role of bar domains in immune cell migration. And so the first big challenge is that actually there is like a ridiculous amount of bar domain proteins. Why is my slide not moving? Hola. There we go.
It did, yeah.
There we go. So there's actually a really, really large variety of bar domains. This is not even all of the ones that are expressed in HL60 cells. And so downscaling this to like a couple of candidates that we can actually experimentally tackle was not an easy task. And so what we decided to do was actually to run an RNA sequencing experiment between the undifferentiated blasts. These are cells that we can keep in culture. They basically hang out, sit around, being round and doing nothing, or our differentiated cells after 5 days of terminal differentiation. And so basically what we were looking at is a condition that has very low migration, very low lamellipodia dynamics, and a condition that has very high migration and very high basically a curvature rich versus a curvature poor state. And so when we did this and we looked at all the bar domain proteins, one of the top candidates was a protein called SNX33. And so this looked like an interesting candidate. There was very little information out there. And so the first thing that Eva did was actually to tag it with GFP and to express it in our cells and to look at where it localized. And what we saw is that it has, do you see my mouse? Yeah. What we saw is that it actually has this pretty cool pattern where at the upper planes of the cell, it localizes at the ruffles. And at the bottom planes of the cell, it localizes a little bit everywhere. And so this real everywhere, but with a clear accumulation at the leading edge. And so with this pattern, we decided to go forward and look a little bit more in detail in this protein. And so we were lucky enough to have a structure that another group had actually put online. And so we could get this PDV domain and we could try and guess how does this protein actually do its job. And so what we saw is that it has a lot of like very positive residues in this particular phase, which very much suggested that it would bind to inward curved membranes. And so we did MD simulations on buccal membranes with various lipid compositions, with various levels of buckling. And what we could see is that it always binds inward curved membranes, and that it does so mainly by electrostatic interactions, not so much by having like an amphiphytic helix that inserts anywhere. And so actually what you see here is that the maximal insertion is on the order of like 4 angstroms. And so this is not even going to touch the tails of those lipids. It's really only going to sense the lipid heads. And so, as I said, we did these MD simulations on these differently curved membranes, and we allowed the cells to basically choose where to go. And what we saw is that unequivocally, they would always bind to inward curved membranes. This is the PX bar with a preference for actually not too curved membranes. So the curvature of radii was not ridiculously small like it is for other bar domains. And so then Eva went ahead and actually used a lattice light sheet microscope, as we saw before. But in this case, she did dual color imaging to image both the membrane and SNX33 at the same time. And what we could see is that even though it does accumulate at the leading edge, it's actually really excluded from the tips. And so we see this either in ruffles, but also at the tip of the bottom lamellipodia, where SNX33 actually does not overlap with the membrane. And so we thought, okay, this looks like a cool protein. Let's go ahead and study it a little bit further. And so what Eva did is generated a CRISPR-Cas knockout at the blast level, right? So you can do this in blast stage because you will be able to keep them. They're not terminally differentiated. We then made sure that they actually differentiate properly, and so that the phenotype that I'm going to show you today is not the result of a defect in differentiation. And so we then went ahead and looked at how do these cells migrate. And so the first thing is we plated them on glass substrates, we allowed them to migrate, and we imaged them with turf microscopy to be able to characterize their cell spread area, but also their leading edge size. And what we saw is that both of these are actually increased in the knockout. Eva went ahead and looked at their ability to polymerize actin, right? Like a larger leading edge is kind of telling you actin polymerization is happening a little bit wacko. And so what we did here is actually by FACS, we can measure polymerization of actin upon a chemoattractant burst. And so what we could see is that indeed you have a higher amount of actin polymerization. And so Eva did these very careful experiments, co-immunoprecipitating whatever comes out with this bar domain, but also with their different domains. Bar domain proteins very often fold like a pretzel. And so they have this auto-inhibited form where all the domains are kind of like mixed around. And so what she decided to do is do the co-immunoprecipitation of the whole protein, but also of the two half, one separated from the other. And what came out with it, actually surprising, was several components of the ARP2/3 complex, but also capping protein. So really suggesting that it can regulate actin polymerization directly by regulating the activity of those proteins. And so if I tell you more actin polymerization and larger leading edges, and you know anything about the work that we've done, this is screaming membrane tension, right? It's like you basically polymerize actin, you push the membrane further. And so we had to look at membrane tension, and very nicely we saw that indeed those leading edges are larger and they can generate higher forces. And so that the theta values are actually higher in the knockout. And very nicely, they can be recovered by overexpressing SNX33 GFP, really telling us that our knockout effects are indeed specific to this protein. It's not some sort of like side targeting effect of CRISPR, which we all know exists. And so we wanted to look further at those leading edges. These look like a really interesting phenotype. And to do so, we use scanning electron microscopy. And so here, we can basically look at the ruffle morphology of those cells. And what we saw is not super intuitive from these images. Actually, Eva had to put a lot of work to properly quantify this. But actually, what we saw is that the wavelength of these ruffles in between each other is changing in the knockout versus the wild type. And so ruffles, this wave-like behavior that generates the ruffles is actually generated by the wave complex, which is the only nucleating promoting factor upstream of the ARP2/3 complex in this sense. And so the wave 2 complex gains its name by the fact that it localizes in waves. And so this was work of one of my postdoc advisors, Orion Weiner. And his lab actually showed that if you generate a knockout of several of its proteins, you actually get 0 leading edges. You get this like, I don't even know how to call these cells. They have this kind of like philopodia, pseudopodia, whatever you want to call it. And so really this nucleating promoting factor is the key one. And so we decided to take a look at what's happening with the wave 2 complex in our knockouts. And to do so, we fluorescently target HEM1, one of the components of this complex. And we saw that we have more wave 2 at the leading edge, and actually these individual patches are larger. And so basically when you don't have SNX33, you somehow end up recruiting more wave with bigger patches that leads to the change in the effective wavelength that I showed you before on the SEM. And so what is this good for, right? After all, this is a cell migration seminar. So we looked at migration. And first, we just looked 2D, right? We know we have these cells, they have larger leading edges, they have more wave 2, they actually migrate better. So if you look at speed, they're faster. If you look at the directionality, they migrate more straight. So what do you want this protein for, right? You are kind of like, in all parameters that we could look in 2D, they were better. But 2D is not the whole reality, right? As I told you, these cells have to avoid obstacles to do their job. And so we decided, with the help of Michael Sixt, the IST in Austria, we decided to challenge them. And basically what we did is we like offered them these bifurcations where they basically have to choose one of the pores. And so here, basically what we were trying to do is we were trying to see, can they adapt when faced with an obstacle? And what we saw is that indeed they're not that good. So once they face an obstacle, they really don't know what to do, right? They don't have a very good adaptive behavior. And so this type of microfluidic devices had been shown by the Sixt lab to show the role of the nucleus. And so we needed to rule out that the problem of SNX33 knockout cells was indeed with their nucleus. And so we did this by measuring nuclear stiffness, and indeed we could not see any effect. So really telling us that their decision time, that their inability to choose properly is very likely due to a defect in cell migration at the leading edge. And so we wanted to explore this contact inhibition of locomotion defect, basically. So in many cell types, what has been shown is that actually when two cells bump into each other, they actually go away from each other in a response to this contact. And so indeed neutrophils do that. And this is actually a very powerful way to spread through larger areas. You basically, every time you contact each other, you will go in opposite directions. And so we wanted to explore a little bit better this contact effect. And so the first thing that we did was actually to quantify what happens at contacts. And so perhaps intuitively, what happens is basically that the two surfaces flatten. And so we use electron microscopy to basically look at the curvature at the leading edge in freely moving cells versus cells that had contacted. And indeed, what we saw is that there is an increase in flatter areas when the two cells contact each other. And so we could further show that this is not due to an artifact in fixation because of the electron microscopy, because we saw the same actually also from lattice light sheet movies. And so very interestingly, what we see is that SNX33 localizes at these contact areas, and it has a very nice avoidance with the wave complex. And so this really told us, is this protein gets recruited to these flatter areas that I just showed you that are formed. What happens when two cells don't contact each other? And so in the wild-type case, I just show you SNX33 gets recruited to the contact, and actually the contact doesn't last that long. Basically, the two cells touch each other, go away from each other. When SNX33 is not there, the two cells touch each other, and they keep on migrating on top of each other because nobody tells the wave complex, hey, go away. And so to really make sure that this was indeed feeding back to the wave complex, which can get annihilated both within cells, but also in between two cells contacting each other, we decided to measure wave dynamics before and after contact in our wild type and in our knockout. And we saw, as expected, from the fact that their contact circumference area is massive and basically doesn't go away, is that indeed these two cells, they contact each other, they touch each other, they don't really sense that they're touching anyone. And so they basically keep on trying to migrate on top of each other, which is not very efficient when you have an obstacle, and it's not very efficient to migrate in complex environments. And so what I hope I have convinced you today is that sensing the plasma membrane actually allows cells to circumvent obstacles. And that this is like, in a way, it's a very simple, but also very powerful way to read your environment, right? You basically are reading out indentations directly inhibiting actin polymerization locally. And so we think that SNX33 couples membrane curvature to actin polymerization. And we also, I hope I have convinced you that this is a very, very effective object avoidance mechanism in complex environments. And so with this, I want to finish my talk by thanking Eva, who was really the absolute pioneer of this project, also Marianne, who did all the electron microscopy, and our collaborators, which one way or another made this story possible. And you for listening.
Thank you so much. Claire will come to you. I think there is a question from Robert Insal, which I'm missing somewhere. Maybe you go ahead. I'll look for Robert's question. It got lost in my chat. I'm sorry.
Hi, Alba.
Yeah.
Hello.
That was a very, very nice talk.
Thank you.
Fine.
But it didn't go the way that I expected, because you start out talking about bar domains sensing curvature, and then you tell us that it goes to the flat place. Is it an I-bar?
No, it's not an I-bar, but it's a bar domain that actually doesn't like that much curvature. And so if you compare it to other bar domains, it likes inward,
Flatter.
but not too inward, flat
Okay.
curvatures.
Okay, that was my, but it's a lovely story. Very, very nice.
Thank you. It's nice to see you, by the way.
You too.
Awesome. Thank you, Claire. I found Robert's question. So he's asking, could SNXX define the boundary between hyaline and non-hyaline at the lamellar port?
Sorry, can you repeat that?
Yeah,
If
so
it
he's
defines what?
So he's asking if SNX33 defined the boundary between hyaline and non-hyaline at the lamellar pod.
We don't have enough resolution to tell you this. So I really wanted to do gold labeling in fixed cells to be able to really look at this. The antibodies are really not good. And yeah, in the end, we didn't explore. I guess we could also take some live imaging we could also use some live imaging to address this, but we have not looked at this level of resolution. I don't know.
Good DIC would show it wonderfully. It's just in the right place, so it would be really interesting. I've always wondered what does that.
Cool. We can explore it further. Thank you.
Fantastic. Questions from Prabhu. Says, with this localization of the SNX33, have you seen recruitment of other adhesion proteins? And is membrane tension using tethers is measuring cortical tension, or is it measuring lipid layer tension?
So I'm going to answer the second question first, and then maybe you have to repeat the first one because I will forget. So by pulling tethers, we can pull tethers two different ways. One is a static tether pulling. We pull a tether, we hold it. Basically, this is what you do with magnetic tweezers, and then it breaks whenever it breaks. This is what we call static tether pulling. And this measures a combination of both in-plane membrane tension of the lipid bilayer and membrane to cortex attachment, but not cortical mechanics. Or we can pull tethers at different speeds, fit a mathematical model, and obtain only this membrane to cortex attachment parameter. And so the data that I have shown you today is from static tether pulling. And so here we have both a combination of the lipid bilayer tension plus this attachment, this surface viscosity that comes from the attachment to the cortical actin. And the first question I forgot, so I will.
Yeah. Prabhu is asking, with the localization of the SNX33, have you seen recruitment of other adhesion proteins?
Oh, no, but we also haven't looked. What I should also say is that neutrophils are ultra awesome. And actually they manage to polarize even in suspension. And so I don't think adhesion is really high on those cells. This is also something that other people have very nicely shown, nor that they need so much adhesion to actually migrate.
Amy Boyd.
Yeah. But we haven't looked. So the answer is I don't know.
Okay. Next question is from Pamela, which made me smile. Says, I know this is cell migration, but have you checked endocytosis?
Yes, we have. And I don't remember. So we have several, Eva has worked with several bar domains, proteins. And so I remember we have a story on SH3BP1, which I'm sure is affected. For SNX33, Eva, are you there? If you're there, just save me here. Maybe she's not. She's not. I don't know. But yes, I don't remember.
It's okay. We can connect you with Pamela if she wants to connect with you.
That's your prerogative as the PI to start forgetting some things. Rachel Moore is
me senior. You people call me a senior PI, so I don't know.
Yeah, that's right. You have a senior moment. Rachel Moore is asking if you've looked in 3D at all.
No, this is something that is on my to-do list in my wish list. We actually have now set up very nice 3D motility assays in collagen gels, so we definitely should do this. What I can also tell you is that quantifying things in 2D is much easier than quantifying things in 3D. And so there was also a little bit this barrier. But now we have good pipelines for a different project in the lab, so we could probably benefit from those.
Awesome. Next question is from Lara C. Saying a really nice story. Did you check about SNX33 in vivo, like in cell migration? Do you know anything about it in vivo?
No, and this is a question that keeps coming back. Maybe she's one of my reviewers. No, I'm kidding.
No, I'm not. No, I'm not.
We have not looked. No, we also are not, I guess, equipped to do some of these experiments. But no, we have not looked in vivo. Thank you.
Great. By the way, everyone, if you do want to ask your question yourself, you can raise your hand and then you don't have to listen to us. From Vera Jansen as wondering about the membrane phospholipid composition. She's working on a homologous protein SNX9, which has an affinity for PIP2 membrane. So she's wondering if there's anything known about SNX33 in this context.
So not much known, but what I can show you, and I have some extra slides. Here it is. So what we've done is what we did is we isolated plasma membranes of these cells. We ran lipidomics, and then we did the MD simulations on lipid membranes that have the composition of those cells. And so what you see here at the beginning is basically what I gave the MD people. Basically, this is the composition that comes out of this lipidomics. We only have plasma membrane isolate, so I cannot do leading edge versus trailing edge versus nothing. Okay, this is like the average of what the cell comes out. And what you see is that once you run these simulations over time, you have a very clear accumulation of POPIs. Yes, exactly. And so you see them here labeled in lila. And so basically they end up representing around more than 20% actually of the, even though they are a small fraction in the beginning, they end up representing a large part of what's bound to this particular monomer, dimer, sorry, but not, we don't have aggregates or other things. I hope that answers. Yeah. And so maybe another way to look at this, just because these slides are not, you see this in like it's the same concept basically. It's basically the amount of time that a single molecule is bound to this particular lipid, and you can see that the traces on the POPI are so much longer because they basically kind of get trapped within the molecule.
That's awesome. Next question is from Adriana asking, what about the interaction of white blood cells with endothelial cells? Do you think that there is a regulation of SNX33? It's all going towards the in vivo,
Yes, absolutely. So, I mean, the cool thing about this project is that Eva did both experiments on microchannels, which are fully inert, right? But also with other HL60 cells. And in both cases, we see the same, right? These cells basically don't choose very well, or they cannot, they have a hard time navigating this obstacle. And so I think this obstacle is in a way physical. It doesn't matter the nature, the chemical nature of that barrier. And so I think it would definitely play a role in transcytosis. But again, this is me saying it without having any data to support that.
Data Shmeda. We have two questions from Anna, and then Olivia, we'll get to you next. So Anna's asking if you've explored in your knockout cells where the centrosome is localized when they've lost directionality.
We have not, but for a different project in the lab, we have set up really good pipelines to do this. This is definitely something we should check. Yeah, no, I don't have an answer, sorry.
Great, and somewhat related, if you have a chemoattractant, is the SNX33 polarized at the front?
Yes, so this is what we see. All the movies that I have shown you today are on a homogeneous bed of FMLP, of chemoattractant, and then we see this enrichment at the leading edge. Yeah.
Olivia, if you want to go for your question.
Yes, hello. So we had published some times ago that in fibroblasts, you also have a system that senses collisions, and that's a system that includes SRGAP2, and SRGAP2 has a bar domain, so it senses curvature, but it also responds to the slit-robo gap system. And so I've shown that in your list, SRGAP1 was the second in your list, and so I think it's interesting. It's more a comment, but maybe it's interesting. We could imagine that there's two collision system, one which is a cell-cell collision system, and this SNX9 would be I don't know, an object collision system or something like this. But I'm still amazed that this SNX9 can talk to a wave like this without going to rack, but that's because I'm obsessed about rigid pieces. But yeah, that's just one comment.
Yeah, thank you. I mean, I don't remember really about SRGAP1, but SRGAP2 is actually sensing outward curvature. Am I right?
Yep.
Yeah, and so what we think is that it's kind of cool that you have both outward sensing and inward sensing, and that somehow these things can either positively or negatively regulate actin polymerization. This is really, I think, the power of bar domains, right? I mean, it goes back to the question of Claire. These guys can sense any curvature. There's going to be one of the family members that can do that. And so this feeding back into the cytoskeleton is an extremely powerful way to regulate motility, differentiation, whatever you want.
Super. Thank you.
Thank you.
If I could, I wanted to ask a quick question because I don't think there are other questions. There are a lot of compliments saying really cool talk, by the way, on the chat.
Thank you.
So I had a question about really actin polymerization, which you were talking initially about, and maybe I didn't get it. What's really the role of SNX33? I mean, I understand later you're talking about the avoidance of wave complex, but how does it regulate polymerization, and does this avoidance at the same time?
Yeah, so we can see that it negatively regulates actin polymerization. This we see in various ways, right? We have larger leading edges. We have actually more amount of polymerized F-actin polymer when we do chemotactin assays and we look
So
at
do you
it.
think it's a competition thing of some sort that?
No, it inhibits actin polymerization. So I think what happens is you basically crash one cell with another, right? And then you locally recruit SNX33 to that interface, and that inhibits actin polymerization. And so wave and the actin polymerization machinery is going to go to wherever there's no contact. And in such a way, you basically migrate away from each other.
Yeah, that's what I meant by the competition. Like it goes so close to the membrane that everything else is just, it's a spatial thing rather than a signaling thing.
I would not go that far as to state this unequivocally. I think, I mean, from the co-immunoprecipitations, we see that it interacts directly with ARP2, several components of the ARP2/3 complex and capping protein. And so it could very well directly inhibit polymerization in that way that then has interesting feedback mechanisms with wave. And so there's more to be explored within this process.
Oh, sounds really cool. I think I took everything, right, Jen? Did I miss anything?
Yeah, if we missed your question in the chat, please let us know. But otherwise we'll start to wrap things up. And of course, if people have any last burning questions, please do ask them now. Otherwise, I'm going to go ahead and stop the